This lecture is going to focus on Chapter 9, Chromosome Variation. It's a really poor name for the chapter, actually. It makes it sound like things that chromosomes are doing that makes them varied. Nothing could be further from the truth. We're actually going to cover the types of broad-range, gross, large-scale, I don't mean gross, disgusting, I mean gross like big picture, large-scale mutations that occur among chromosome molecules. We have a whole separate lecture dedicated to the individual DNA mutations that occur on chromosomes, but today we're going to talk about these large-scale mutations that change overall chromosome structure. So we'll begin talking about and defining these chromosomal mutations that occur on the large molecular level. And they'll break down into being duplications, deletions, inversions, and translocations, and we'll thoroughly define and describe all of these different types or flavors of chromosomal rearrangement mutations. We'll end the lecture talking about aneuploidy. That's when you have one or two chromosomes too many or too few. And polyploidy, which is when you have one or two sets of chromosomes too many or too few. So overall, chromosome number is important. And what we mean by that is that the number of chromosomes individuals of a species are supposed to have tends to be the number of chromosomes individuals in that species does have. And for most species, all individuals, regardless of their gender or their subtype, have the same number of autosomal chromosomes. Sometimes there's differences in the sex chromosomes, as we saw for flies, but autosomal chromosomes tends to be a conserved number. And all autosomal cells of individuals of that species tend to have the same number of chromosomes as well. This is because the number of chromosomes is a hallmark feature of all species. All species have evolved to have the right genes, of course, but more importantly, or just as importantly, the right numbers of those genes, the right balance of the different genes to make that organism thrive. So you may remember a while ago when we were talking about the X chromosome especially, we talked about the balance of genes, the balance of alleles being key. That's in fact why the X chromosome is inactivated, why bar bodies exist. Bar bodies exist and X inactivation exists because males of human species have only one functional X chromosome, and we had to preserve that balance in females too. So we deactivated or tightly wrapped up and made inaccessible a female's second X chromosome, and that gave rise to a bar body. So this theme of balance being key for genes and alleles and chromosomes has been going on in this course since almost since the first lecture. Life is always striving to keep its balance, to keep its complex systems in balance. This isn't just true for chromosomes, this is true for all life systems. You probably heard the word homeostasis in 8th grade, this idea of steady as she goes, this idea of balancing different life systems in a complex organism, and this balancing of chromosomes is no exception to that. But sometimes things go wrong. And when things go wrong for DNA, we refer to that as mutations. So there are types of mutations that affect chromosome number and screw up this balance, screw up this chromosomal homeostasis. Because the balance is being perturbed and because the balance is so important, these mutations tend to be very severe and detrimental to the organism. There are also mutations that affect chromosome structure on a large, wide scale, not small mutations of a small subset of DNA sequence in a single gene. We'll talk about that in another lecture. Large mutations that affect the structure of the entire chromosome. Together, these types or classes of mutations, those that affect chromosome number and those that affect large-scale chromosome structure, are referred to as chromosome mutations. I'll also refer to them as chromosomal rearrangement mutations to kind of make that distinction. And that's what this whole lecture today is going to talk about. So before we can actually go down this road of changing chromosome structure and changing chromosome number, we should have some idea of what the right way should be. What is the right chromosome number, which we're familiar with already? And more importantly, what's the right correct chromosome morphology or structure? And we've talked about some of these concepts already. We already know what a centromere is. I hope we talked about centromeres sort of extensively in the mitosis and meiosis lectures. We briefly mentioned telomeres as well. We use the analogy that telomeres are like the caps of shoelaces. They keep the chromosomes from fraying and shortening at their ends. That's very much true. Based on these two structures, centromeres and telomeres, chromosomes are divided up into 4 morphological classes, and by that we mean 4 different shapes or structural shapes that chromosomes can take. They are metacentric. I'll show you a picture in just a second to kind of drive the point home, but to define them, they are metacentric. This is where the centromere is located pretty much in the middle of the chromosome, giving 2 arms, the top arm and the bottom arm, of relatively equal length. Submetacentric, where the centromere is displaced just a bit towards one end or the other, creating one arm that's a bit longer than the other. Acrocentric, here the centromere is very, very close to one end, creating one very, very long arm and just the smallest hint of a short arm on the other side of the centromere. Or telocentric, remember telomere and centromere, telocentric is when the centromere is at the cap of the chromosome, pretty much resulting in just one long arm and no other arm on the other side of the centromere because the centromere is at the end of the chromosome. And all of these things could be said 2 arms if we're subsequent to interphase and the chromosomes have been replicated and we're looking at sister chromatids. These are sister chromatids that we're looking at here. That's why I bring it up, not to confuse it. We're looking at 2 recently replicated chromosomes. So we have 2 sister chromatids connected. Keep in mind, this is a sister chromatid. This is a sister chromatid. They are physically connected to one another at the centromere, but we are still looking at one defined chromosome. And we're pretty familiar with that terminology, I hope. And here's what I just said, just in picture form, which is always, I think, a lot easier to see and understand than words. Metacentric, your centromere is right in the middle. Submetacentric, your centromere is off middle just a bit, but you still have a very definable short arm as well as a definable long arm. Acrocentric, where the centromere is very, very close to one end, but still short arms are apparent. And telocentric, where the centromere is so close to the end that there's really no other short arm that can be detected. Those are the 4 morphological classes of chromosomes that we are aware of. And if we look at this karyotype of human chromosomes, we could see that our own chromosomes naturally come in all flavors. I would call this metacentric here, for all intents and purposes. This is certainly submetacentric, I think. This is acrocentric, I think it's fair to say, very short arms. And then somewhere floating around here I saw a here we go, here's telocentric, right here. So, different morphological classes. We're characterizing the chromosomes just on their shape, on their morphology, and we'll come back to some of these ideas as we go on later in the lecture. So, if that's what chromosomes should look like, what are the different types of chromosome mutations that we might be faced with? In general, there are 3 broad categories of these types of large-scale chromosomal rearrangement mutations. The first are the chromosomal rearrangements. Here, the overall chromosome number is unaffected. And so we're supposed to have 46 chromosomes, and if we have chromosomal rearrangements, we still have 46 chromosomes, but large pieces of chromosomes move. These mutations, they alter the chromosome structure because now we have pieces, large chunks of chromosomes moving all over the place. The second broad category of chromosome mutation is aneuploidy. I already loosely defined it in the first slide of the lecture, but aneuploidy is when we change the number of chromosomes up or down by 1 or 2 or 3. So we have the loss of a single chromosome, so instead of 46 we have 45. We have the loss of 2 chromosomes, so instead of 46 we have 44. We have the gain of 1 or 2 or 3 chromosomes, so instead of 46 we have 47, 48, 49, etc. Down syndrome is an example of aneuploidy. So is Klinefelter's, which we talked about in our sex chromosome, sex differentiation lecture. Turner syndrome also we talked about then. These mutations alter the chromosome number. Polyploidy, also I introduced on that first slide. This is when we're talking about sets of chromosomes being added or taken away. So here, if we're supposed to be 2n, a polyploid might be 3n. By 2n, we mean we are supposed to have 2 copies of 23 chromosomes from each parent. That gives us our 46. A polyploid or a triploid individual of the human species would not be 46, they would be 69. Let me see if I did that right. Yeah. So we have 3 sets of 23 chromosomes. giving us 69 total chromosomes. That would be an example of polyploidy. And here is a karyotype of a polyploidy individual who did not survive. I'll come back to that later on in the lecture. This would not be a viable human being. But you can see here that we have a triploid human being. 3 sets of chromosomes, 69 total. Humans don't survive as polyploids. Some animals do, even that's a rare exception. It's pretty common in plants, and we'll come back to that later on in the lecture. These mutations also alter chromosome number, because if we're supposed to have 46 and we have 69, obviously we've changed the chromosome number, but it's different than aneuploidy because we're not changing by 1 or 2 chromosomes, we're changing by entire sets. So this figure from your textbook, I think, does a pretty good job of driving home these 3 broad classes. We have our chromosomal rearrangements, where we have large segmental changes in our chromosomes. This is showing a subtype called a duplication. We were supposed to have 1 set of CDE regions, but that has been duplicated. This is just one example of a chromosomal rearrangement. But we can see a few things. One, we've changed the overall structure of a chromosome from what it should be to what it is now. But more importantly, we haven't changed the overall chromosome number. This individual, although mutant, does not have an extra chromosome. They still have 46 chromosomes total. Aneuploidy is when we have a 1 whole chromosome added or taken away, so we're changing the chromosome number by 1 or 2 or 3. And polyploidy is when we're gaining entire sets. So we have an extra chromosome for every single pair in the chromosome set. So now we change by units of chromosomes. We change by units of chromosome number from 23 to 46 to 69. And that's enough of that. So how do we recognize these? Obviously, these are going to be very detrimental. Obviously, we need some way to diagnose these issues, to spot them, to track them. And so how do we see chromosomes so that we can measure and detect chromosomal rearrangements, polyploidy, and aneuploidy? Well, we do it primarily through karyotypes. We've talked about karyotypes a few times in this course already without really defining it. I'm working on the assumption that karyotypes have been defined for you in other classes. Here is a human karyotype of a male, obviously. Hopefully it's obviously, because we have 1 X and 1 Y chromosome here. Usually to make a karyotype, you harvest some rapidly dividing cell from the individual. Those could be white blood cells, bone marrow cells, any adult stem cell that is rapidly dividing will do. You treat those cells, you have them divide for a while, you get them undergoing mitosis at a pretty high frequency, and then you treat them with a chemical that blocks all of those cells in metaphase. So all of those cells get stuck in metaphase of mitosis. And think about what happens in metaphase. In metaphase, all the microtubules are attached to the chromosomes. The chromosomes are lined up at the center of the cells. Sister chromatids are still connected, right, because we haven't gone to anaphase yet, but the chromosomes are very, very compact and very dense, easy to see under a light microscope. These cells are then fixed, meaning they are locked in place with something like formaldehyde. They are lysed or burst open, spilling those chromosomes out into the media of the experiment, and then the chromosomes are spread on a microscope slide. Those chromosomes are then photographed on the microscope slide in their random orientations, such like we saw in this slide here. And then in the old days, literally, by hand, the picture would be cut into pieces, and the chromosomes rearranged in some logical order, as we're used to seeing in a karyotype. Now, of course, there are computer programs that do this for us and isolate these chromosomes, take the images of them, and line them up for us nice and easy. But in any event, however it's done, the chromosomes are arranged artificially, visually, in size order, and therefore in number order. For all species, the largest chromosome pair is always given the number 1. The second largest, number 2. The third largest, number 3. All the way to the smallest chromosome pair, which is given the last autosomal number of the set, and then the sex chromosomes are always kept separate from those. So we have 22 autosomal chromosomes for a total of 44 autosomal chromosomes total, and they are arranged in size order, with chromosome pair number 1 being the largest and chromosome pair 22 being the smallest. Now that tells us how we get them and how we see them, but it certainly doesn't tell us how we visualize them in a way that we can easily measure what's going wrong. To do that, we need chromosome stains. And there are a number of different chromosome stains available that make chromosomes easy to see microscopically, and depending on which stain you use, you can highlight or visualize different parts of the chromosome. I'll give you some examples here. There's Geisma stain. Geimsa stain. Geimsa stain reveals what are referred to as G-bands. The stain sticks more to areas of the chromosome that are rich in adenines and thymines. It sticks less well to areas of the chromosome that are rich in guanines and cytosines. These are the A's, T's, G's, and C's that make up the DNA sequence. And so when you use this stain, you highlight these bands, these striations, these stripes in the chromosomes, as you see here. And these stripes become telltale markers of where you are on the location of the chromosome. These striation patterns are how we know what part of the chromosome we're looking at. And I'll give you examples of that later on once we go through some of the mutations that we can encounter. There's also quinacrine mustard stain. Quinacrine mustard stain is very, very nice because it's fluorescent. It can be used under UV light, so it makes those chromosomes very, very visible. And they light up Q bands, different than the G bands. The Q bands also light up DNA based on their AT and GC content, just does so a little bit differently than the Geimsa stain, and so the striation patterns are quite different. Geimsa stain has been altered or modified, so we have a variation of this staining that selectively visualizes only centromeric DNA. We call these C bands for centromeres. This often gives a very, very pretty staining, and you can see here all these dark stained regions are showing us the centromeres. So when you use this variation of Geimsa stain, you can very easily spot your telocentric, your metacentric, your submetacentric, and your acrocentric chromosomes, because you can easily spot where on those chromosomes the centromeres are. So these stains almost become a toolkit, almost like an artist's palette, that allow you to stain your mitotic chromosomes in any way that you see fit for your experiments, depending on what it is you want to see. Do you want to see general striation patterns under a normal compound microscope? Well, you better use Geimsa stain, because that's what that's going to allow you to do. Are you doing fluorescent microscopy and you want your chromosomes to be visual under a fluorescent microscope? Well, then you should use quinacrine. Are you looking just at the centromeres and all you're interested in is the placement of the centromeres? Then you should use this variation stain. So depending on what you want to see, it dictates what stain you should be using. The point is, you can see your chromosomes under microscopy, and more importantly, you can see the substructure of those chromosomes. You can get some idea of what types of sequences are present in what locations of each of these chromosomes. So with that technology, we can recognize many different types of chromosomal rearrangements using karyotypes. And so we're going to talk about those different types of chromosomal rearrangements. We'll stay away a little bit from what we see on karyotypes. I just want you to be comfortable with what these words mean and what types of mutations they result in. We'll cover the 4 basic types of chromosomal rearrangements now. These are duplications, deletions, inversions, and translocations. And for each of these explanations, we're going to use this notation to represent just random, everyday, whatever kind of DNA sequence you want to imagine on the chromosome in question with a centromere. So what I really mean is this is a very, very long chromosome that's represented here, made up of nothing but DNA. This is a metacentric chromosome, so the centromere is pretty much in the middle. And we are just kind of saying, well, this chromosome is made up of regions or segments of DNA. We're not really interested in the sequence of that DNA right now. We're just interested in them as regions, and we're going to give these regions of DNA each their own distinct letter, just like a name that we can refer to the region as. So when we use this donation style, we're talking about a true DNA chromosome with a centromere right in the middle in this case, with DNA sequences or DNA regions on either end. That's all we mean. And so we can use this notation to represent these different types of mutations in a pretty simple and straightforward way. And here's the perfect example. When we have a duplication, a chromosomal duplication, that's when we have a part of the chromosome duplicated or doubled. There are two types of duplications: tandem duplications. Tandem, one right after the other, just like soldiers marching in tandem. We can have duplications occurring in tandem, one right behind the other. And using this notation, this is what a tandem duplication would look like. It's easy to spot which region has been duplicated, and it's easy to see why it's in tandem, because it's one right after the other. And so this is what we mean when we use this notation in this way. So this is a tandem duplication. The opposite of a tandem duplication is a displaced duplication. Here we have the duplication, but it's not one right after the other. The duplication is somewhere else, anywhere else, and here is where the duplication happens to be in this notation of a chromosome. So we have duplicated region EF. EF still exists where it was supposed to be. It has been copied or duplicated, and that duplication has gone somewhere else on this chromosome, giving us displaced duplication. There are also reverse duplications, where the duplication region is in tandem, but in the opposite order, and so we would use this notation to represent that. This could have been a tandem duplication if it was EF-EF, but instead it's FE-EF. That's the reverse. That's the reversion, and so we call this a reverse duplication. So pretty easy, I think, pretty straightforward, hopefully. So what are the effects of these duplications? And for each of these chromosomal rearrangement mutations, this is the structure we will have. We will define the rearrangement so that you understand what's actually going on the DNA molecule, and then we will discuss the effects, the potential effects of these mutations on the chromosome and sometimes on the individual themselves as well. For all of these chromosomal rearrangements, individuals can be homozygous or heterozygous for them, such is the case for duplications. You're homozygous for the duplication if the same duplication occurred in the same way on both chromosomes for that pair. In other words, if chromosome 1 from your dad had a tandem duplication and chromosome 1 from your mom had the same tandem duplication, then you're looking at a homozygous individual for that duplication. But much more likely, of course, is heterozygosity for the mutation, where the duplication in this case happens on one chromosome of the pair, but not the other. When an individual is heterozygotic for a duplication, the first set of problems we see arise during prophase 1 of meiosis. You see, in prophase 1 of meiosis, we're supposed to have synapsis. We're supposed to have homologous chromosomes line up right next to each other, side by side, almost zipping up together with all of their homologous regions kind of touching and feeling one another, getting ready for crossing over. But once we have a duplication event, those two homologous chromosomes, if they're heterozygous, can't zip up perfectly with one another anymore because they aren't perfectly aligned, as shown in this diagram. In order to have that synapsis occur and have that zipping up, one of those duplicated regions must loop out, allowing the two chromosomes to align with one another. That looping out puts a physical strain on the DNA molecule and could result in breakage of the DNA molecule. Duplications may sometimes have significant effects on the individual's phenotype. This goes back to the initial idea of homeostasis with chromosome number and gene number, and the balancing of all the genes, having the right genes in the right amount, and not having too many or too few. When we have too many or too few genes due to chromosomal rearrangements, we refer to that as an unbalanced gene dosage. Why? Because we're supposed to have a certain dosage of our genes, or a certain amount of our genes, and if we have the wrong amount, then we are unbalanced for that gene amount. So unbalanced gene dosage, the wrong or unbalanced amount of genes in our genome. We as human beings are evolved to have two alleles for each gene, no less, no more. One from mom, one from dad, each allele doing whatever the hell it is they were supposed to do. Be codominant, be partially penetrant, be completely dominant or recessive. Whatever they're supposed to do, that's what they're supposed to be doing. And to go back to that very, very first analogy that I hit you guys with, we're supposed to have two machines pumping out brown hair. Not one, not three. Our factory has evolved to have two. When we have more than two genes working, more than two alleles, more than two machines working on our factory floor, And this could easily occur due to a gene duplication event. Bad things can happen. Here's a real-world example. There's this bar region on the fruit fly chromosome that is involved in eye development. And the fruit fly has evolved to have two of these genes, two of these regions on it. And when the fly has two of these bar regions on their chromosomes, it develops normal eyes. If you have a gene duplication event, and you have three of these regions, here we see a tandem duplication, we see that the fly's eyes don't really develop correctly anymore. We start to see a smaller eye phenotype. If we have four of those regions, so now we have two duplication events, but the organism is homozygous for that duplication, so at least we don't have any problems during prophase 1, we see an enhanced shrinkage of the eye. Most dramatic if we have 4 copies of that gene, but 3 of them are on one chromosome, so we have 3 tandem duplications on one chromosome, and the other is not. This is going to result in a great deal of looping out during prophase. We see the most severe phenotype, and the eyes are barely there at all. So this bar region, the genes in this bar region, play a role in eye development by restricting the eye formation, not allowing the eye to be too big. But too many of those restrictive eye development genes restricts eye development too much. We have unbalanced gene dosage. The genes that are responsible for making eyes can't overcome this additional restriction, and so the fly develops much smaller eyes. So duplications are often bad. But it's important to point out that duplications also do allow new genes to be born in a way with new and novel functions. You see, when a gene duplicates, especially if that gene was essential for some life-critical function, when a gene duplicates, the new duplicate gene might be bad, might be too many of those genes right now, but now that new duplicate gene is free to be mutated. See, that's an interesting idea. And when this gene mutates, it's now evolving. And while it evolves, this new gene can take on new function. You couldn't do that before the duplication event. You can't tinker with an essential gene. A gene that's essential is essential. You can't mess with that. If you screw up an essential gene, the organism's going to die. So what if you want to give an essential gene a new function? You want to tinker with it. You want to mutate it, try it out, evolve it. Mutate it some more, try it out, evolve it. What could you do? Well, you could take your essential gene and have a new copy of it. Make a copy of it and play with that copy of the gene. Mutate that copy of the gene. If you screw it up, no big deal. You've got the original gene backing you up. But if you can take on a new function, an improved function, now you've got development of the species. Now you've got evolution in a positive direction. It's like your iPad or your smartphone or whatever other technological device you can't live without. You could pop the back off of that device and you could tinker with it, and you might make it a better smartphone. You might improve that smartphone. Right? Chances are, though, you're going to screw it up because you don't know what the hell you're doing. Random mutations are going to, chances are, screw up a gene. So if you really want to tinker with that smartphone, if you really want to try your hand at improving it, but you can't live without it, what makes the most sense? Well, the most sense would be to go out and get yourself an identical copy of that smartphone. Keep one as a reserve in case you screw up the other. But if you make the other better, you've won. So it's this safety net idea, having one gene there that can back you up if the mutation goes bad, but allowing the mutations to try at hand at making the gene better. This type of gene duplication event combined with mutation and evolution allows evolution to proceed forward with little risk to the individual because that original gene is there backing the process up. This is actually what allowed fetal hemoglobin to evolve. Fetal hemoglobin is the direct result of a gene duplication event of the original hemoglobin gene. And now that new hemoglobin gene, that new duplicated human hemoglobin gene, could be mutated and tinkered with and evolved, making fetal hemoglobin possible. So, many of you may be familiar with fetal hemoglobin. Some of you may not. I'm going to quickly tell the story because to me it's an amazing story. If you haven't heard of fetal hemoglobin before, you're going to realize that there's actually a pretty big problem in utero development that you may not have realized already. So, we all know what hemoglobin does, I think. Hemoglobin carries oxygen in the bloodstream. And I think we all have a general sense of where babies come from, and I don't mean the birds and the bees, I mean that babies develop in the womb. There's a placenta in the womb. The placenta is the interface between the fetal blood supply and the maternal blood supply. The maternal blood and the fetal blood come very, very close to physically touching at the placenta, but they never do mix, and this is how the mother gives the fetus nutrients and takes waste out of the fetal blood, and of course processes that waste herself. So this is critical to the development of the embryo. So now I'd like you to think about something. Mama has hemoglobin in her red blood cells. Baby has hemoglobin in baby's red blood cells. And now imagine for a moment that the hemoglobin is the same. It's the same exact molecule, same exact protein for carrying oxygen. So mama takes a breath in. Oxygen latches on to mama's hemoglobin because of that breath she took. That oxygen now travels through the bloodstream. It goes by the placenta. That maternal hemoglobin is carrying the oxygen, and the baby's hemoglobin needs that oxygen. Baby can't take a breath on its own yet. Obviously, it's in utero. But if that hemoglobin protein is exactly the same in the mother and the fetus, why and how in the world would the fetal hemoglobin get that oxygen off of the mother's? Fact of the matter is, 50% of the time it couldn't. It's like two people of equal strength fighting over the same cookie. Two hemoglobins of equal or identical strength fighting over the same oxygen molecule. For every time the guy on the right wins and gets the cookie, there's going to be another time where the guy on the left wins and gets the cookie because they're of equal strength. That's not good for the fetus. The fetus needs a lot of oxygen. And in fact, that's not a detriment to the mother because the mother can breathe. The mother can breathe as much as she needs to get her oxygen saturation levels up. So what happened? Well, way, way, way back when, after a whole lot of inefficient development in mammals, there was a gene duplication event. And the gene duplication event resulted in another hemoglobin gene. So now individuals, mammals, early mammals, had two hemoglobin genes. Hemoglobin is essential. You tinker with hemoglobin, you're going to screw things up. We need hemoglobin protein to carry oxygen. You mess up that system and organisms are going to die. So you couldn't tinker with hemoglobin before the gene duplication event. But once that gene was duplicated, now you had one hemoglobin you could futz with, and the other hemoglobin you could leave alone. That new hemoglobin mutated over time, getting worse, then getting better, getting worse, and then getting better, until it mutated to a point where it was actually stronger than the original hemoglobin was. The new tinkered with hemoglobin was actually better at carrying oxygen than the initial hemoglobin was. This better hemoglobin was then reserved for fetal development. So now the fetus got to express this stronger hemoglobin, and the mother was expressing the weaker adult hemoglobin. Now imagine that same scenario. Mama takes a breath in. Oxygen is loaded onto her hemoglobin. That blood circulates and it goes by the placenta. There at the placenta is the stronger, more powerful fetal hemoglobin that physically yanks the oxygen off of the maternal hemoglobin and gives it to the fetus much more efficiently. Now it's not two hemoglobins of equal strength fighting over a cookie. It's the fetal hemoglobin getting the cookie almost every time. The mother's not at a deficit. The fetus is so small that there's still plenty of adult hemoglobin floating around the mother's body with oxygen on it, because there's not very much of the fetal hemoglobin, but every time fetal hemoglobin needs oxygen, it gets it, because it can rip that hemoglobin off of the maternal molecule, the maternal hemoglobin. And so we have this constant one-way flow of oxygen from the mother's blood to the fetus's blood, from the mother's hemoglobin to the fetus's hemoglobin. Why? Because the fetal hemoglobin is better at carrying oxygen than the mother's is. Why? Because we had this opportunity to tinker with the hemoglobin gene, making a better protein. Why? Because we had a gene duplication event way long ago in the evolution of mammals that allowed this tinkering to occur. But it's important to note that not every story ends as well as the hemoglobin story. It's a crapshoot anytime you do this. Like most mutations of all kinds, this is chromosomal rearrangement mutations as well as the smaller individual DNA sequence mutations. Most of the time, most of the time, mutations, including duplications, are bad for the individual. So most of the time, the individual suffers as a result of the mutation. But without these random mutations, the species would never have the opportunity to evolve. We are all at the mercy of the evolution of our species, and 99.9% of us suffer as a result of the mutations that the species is using to evolve. But for that 0.1% that is bettered, the species is bettered, and that is evolution. Progression of the species at the expense of its individuals. Okay, so what other types of chromosomal rearrangement mutations can we talk about? Well, we have deletions. Chromosome deletions are the loss of a region or segment from a chromosome en masse. This is a large-scale deletion. Going back to our trusty notation, a chromosomal deletion would look like this. Region EF is gone. It's gone. It was removed by a deletion event, and it was destroyed once taken out of the context of the chromosome. If the deletion is large enough, you can see it in a karyotype as a shorter than should be chromosome, missing some of those banding patterns that we should see with our stain if we had a healthy chromosome of appropriate size. Much like duplications, deletions also result in looping out during prophase 1 for the same reason, because we have to have this synapsis zipping up during prophase 1. Here it's the healthy chromosome that needs to loop out, because it's the healthy chromosome that has more DNA than the other, but still we have looping out. The effects of any particular deletion depends really solely on what was on the DNA that was removed. If there weren't really any important genes on the DNA that was deleted, then it's pretty much an asymptomatic deletion, which is very possible. However, if supercritical genes were on that DNA that happened to be deleted, then those deletions could be lethal and the organism might die as a result. So the effects of chromosomal deletions depend solely on what was on the DNA that was deleted and how important was that DNA to the organism. If the deleted region includes the centromere, then the mutated chromosome is not going to be able to segregate properly during mitosis or meiosis. The kinetochore is at the centromere. The microtubules connect to the kinetochore. So you can have a deletion that removes the centromere, and then the cell has no way by which to attach microtubules to that chromosome during mitosis or meiosis. And so the chromosome often gets lost by the cell during cell division because we can't get segregation. Most deletions are highly detrimental when an individual is homozygous for the deletion. That's when both alleles have been removed. So here we have the complete absence of that genetic information in total. But even when heterozygous, deletions cause imbalance because we're supposed to have two alleles, and now we only have one. Also, remember, we are the product of dominant and recessive alleles. Many of us have mutant and defective recessive alleles in our genomes, but we are perfectly healthy because we have a dominant allele to counteract that. If the dominant allele was on a portion of the chromosome that was deleted, now we only have the recessive allele, and we have to get by with that. And that obviously can be problematic. We have unbalanced gene dosage, just as we had for duplication, so just as too much of genes is not good, not enough of our allele number is not good as well. Recessive alleles express more readily when there are deletions. I just hit on that, got a little bit ahead of myself there. So if we have a deletion, then the deleted region behaves like the male X chromosome. We talked about color blindness being much more prevalent in males because males only have one X chromosome. So if they have a defective recessive allele for color blindness, then that's the trait they show. Once we have a deletion, then the deleted region behaves that way too. We only have one present region containing those alleles, and so you express whatever allele you happen to have maintained. There are some very rare human syndromes associated with genome chromosome deletions. I'm not going to go into those here because it just adds a lot of detail that we don't particularly need. But if you're interested in what types of things occur to people when we have chromosomal deletions, by all means see the text. There's a pretty good write-up in the textbook for this. So that's duplications and deletions. Continuing on, we'll talk about inversions. To visualize a chromosome inversion, you have to imagine a segment of DNA literally popping out of the chromosome, spinning around 180 degrees, and then popping right back in to where it started. It's pretty weird. And if you can't visualize that, I'll do it for you here as an animation, so keep your eye on region EF. We really mean it pops out, spins 180, and pops back in. That's an inversion. For this to occur, think about it. The DNA actually had to physically break in two places on either side of the inverted segment. To get region EF out, we had to snip it out and remove it from the chromosome, then spin it around and glue it back in place. That happens. We have enzymes to snip DNA and we have enzymes to glue DNA, so this does happen, and it happens more readily than you might expect. There are two types of inversions, paracentric and pericentric. Paracentric inversions do not include the centromere. What I showed you in this animation is a paracentric inversion. Pericentric inversions do include the centromere. This would be the example of a pericentric inversion. The centromere is included in the inversion event. It's believed that inversions played a pretty active role in the evolution of human beings, and we can see this, we can actually track this. Again, going back to our staining idea, we can take human chromosomes and chimp chromosomes and we can stain them with some of our stains and get those unique striation patterns. If we compare those chromosomes then, we see that humans and chimps do share an enormous amount of genetic identity on the DNA level. Most of our DNA is the same, but what we also see is that the layout or the arrangement of our chromosomes is quite different in its shape or structure. So human chromosome number 4 and chimpanzee chromosome number 4 is nearly identical in its sequence. That is, if you are to look at the sequence of any gene on the human chromosome, it's nearly identical to that same gene on the chimp's chromosome. But at a larger scale, if we zoom out a bit, what we see is that this region in the human chromosome is flipped 180 or inverted from the same similar region on the chimp chromosome. Now, is this red gene in the human chromosome nearly identical to the same gene in the chimp chromosome? Yeah, but it's in a completely different spot because of an inversion event. So you might say, well, how could that be? Why would that matter? Why would flipping the DNA change anything? Well, it does. Chromosome inversions have their own effects. The first is more obvious. If an inversion breaks a gene in half, moving half of that gene to one part of the chromosome and leaving the other half where it was, it's almost like taking a piece of paper and cutting it in half. It's going to lose its meaning. You can't read that page anymore because the two halves of the same page are on different locations. So the gene will be rendered inactive, and that's what we would call a null mutation. The gene no longer functions. If the gene was important, obviously it's going to have detrimental effects if it no longer functions properly. But getting back to the chimp problem, the chimp issue, many genes are regulated, and by regulated we mean turned on when they're needed and turned off when they're not. They're regulated based on their position on the chromosome. So where the gene is on the chromosome allows or helps that gene to be turned on so that a protein can be made from it, or turned off so that proteins are not made from it. Once you change a gene's position on the chromosome, you can also change its regulation, meaning you're now making protein from that gene when you shouldn't be, or you aren't making protein from that gene when you should be, and that often results in bad things. We refer to this as the position effect, where the position of the gene on the chromosome affects its function. Now keep in mind, the sequence of that gene is unchanged. It's just where it lies on the chromosome that has changed. But that change in position affects the regulation of the gene. This is likely what allowed us to differentiate ourselves from the chimps. Even though our genes are very similar in their sequence, they are quite different in their position, meaning they are quite different in their regulation. Individuals that are heterozygous for inversions require some pretty super funky twists and loops in order to allow synapsis. You can see these two chromosomes that have undergone an inversion event. One has undergone an inversion event, the other hasn't. Have to really knot up with one another to zip up properly for for synapsis in prophase 1. It goes without saying that these types of loops and knots often result in chromosomal stress and chromosomal breakage, which then in turn results in chromosomal loss, and this can be very detrimental to the organism that has these cells in it. So those are inversions. The last of our chromosomal rearrangements, the translocations. When DNA moves from one chromosomal location to another non-homologous chromosome, we have a translocation. So now we're talking about taking DNA out, taking a segment of DNA out, just as we did for an inversion. But instead of flipping it around and putting it back where it was, now we take DNA out and put it somewhere else on another chromosome completely. Like taking DNA out of chromosome number 1 and putting it on chromosome 15. To represent this using our notation, we actually have to introduce a non-homologous chromosome. So we're going to move to another chromosome, organized the same way, but made up of completely different sequences. So this might represent chromosome number 1, and this might represent chromosome number 15. This is a translocation. A region from chromosome number 1 is removed and moved to chromosome number 15. Translocations can also occur within a single chromosome, where a region is chopped out from chromosome number 1 and then moved to some other non-homologous place on chromosome number 1, like between G and H. That's also possible. Not as common, but also possible. What we show above is referred to as a non-reciprocal translocation. This is where it's a one-way transfer. DNA was chopped out of one chromosome and moved to another, but that's it. That's the only DNA that moved. You might be able to then imagine in your mind what a reciprocal translocation is. Here is where DNA is removed from two different chromosomes and swapped, so it's an exchange in both directions. We can do this denotion pretty easily using these two chromosomes, and here is a reciprocal translocation. We have a region from chromosome number 1 that moved into chromosome number 15, and we have a region from chromosome number 15 that moved into chromosome 1, a reciprocal exchange. And reciprocal translocations are far more common than non-reciprocal. Now, some of you who are astute and really good at synthesizing the information we've been covering all semester might take one look at this denotion and say, what do you mean that's a reciprocal translocation? I've seen this before. I've seen the exchange of genetic material. I've seen two chromosomes swap pieces, and you told me that was crossing over. You told me that was recombination during meiosis, and you told me that that was extremely beneficial to the organism, never to be classified as a mutation. And I would say, wise one, and don't get ahead of yourselves here. Crossing over and recombination during meiosis is only between homologous chromosomes. Only between homologous chromosomes of a homologous pair. That's where the genetic exchange occurs. Translocations are defined as DNA moving from one chromosome to a non-homologous chromosome, a chromosome of another number. And so that is not the same as crossing over. What are some of the effects of translocations? Well, translocations cause new linkage patterns to emerge. Genes are now linked to genes they weren't linked to before because they're now physically on the same chromosome molecule in a way that they haven't been before. That can change how these genes are inherited. That can change how these genes are regulated because we still have these position effect things to keep in mind. Position effects occur when genes are regulated in a certain way because of where on the chromosome they are found. If a gene is moving from one chromosome to another, we have changed its position, and we can very well change its regulation as a result. Oftentimes, when we have a translocation, because we're cutting the chromosome, we are also having chromosome deletions as well. They often occur simultaneously. We can see this in this example. We have a pretty severe translocation where breakage here on the green chromosome and breakage here on the orange chromosome result in a re-gluing of these two chromosomes to one another. So here is a new chromosome that is made as a result of that translocation. It is much, much larger than either of the original chromosomes were because of where those two translocations occurred. Of course, what's left over, the pieces from the remaining chromosomes also translocate, so we do give rise to this very small chromosome as well, but that chromosome is so small that it's going to contain very, very few genes. It's going to contain very, very little information. And so it's very easy for the cell to lose that chromosome entirely because it is so non-essential. This particular type of translocation is a way by which Down syndrome can occur. It's an uncommon form of Down syndrome, but it is a way that Down syndrome can occur. Getting back to our comparison with our nearest cousins on this planet, chimps, orangutans, and gorillas, our three closest cousins on the primate lineage, all have 48 chromosomes. How many chromosomes do we have? 46. So we're 98% or more similar to these great apes, yet they have 2 more chromosomes than we do? How the hell is that possible? Well, if you look at chromosome number 2 in humans, and you stain it so that you get that very unique striation pattern, almost like a barcode or a fingerprint of the chromosome, we see that human chromosome number 2 is quite large, and we know that because it's chromosome number 2. It's the second largest chromosome in our genome. We compare that to 2 chimp chromosomes of much smaller size, and we see this remarkable similarity in banding pattern. And we say, well, that's uncanny. It almost looks like the long arm of chromosome number 2 in us matches this entire chromosome from chimps. And then we see some conserved banding pattern on the short arm of chromosome number 2 with chimp chromosomes as well, other chimp chromosomes, separate chimp chromosomes. We look at gorilla chromosomes and we see the same conservation. We look at orangutan chromosomes, we see the same conservation. And we realize then, my God, what has happened somewhere along the line of the human lineage is that 2 primate chromosomes underwent one of these translocation and deletion events where 2 smaller chromosomes became stuck together as one large chromosome. That is our chromosome number 2, and the smaller informationless chromosome was lost somewhere along the way because it contained so few genes. So if you take 2 chromosomes and you condense them down into 1, you take 24 chromosomes. Let me get rid of my writing. You take 24 chromosomes in chimps, and you get it down to 23 in humans. Now, if you take the 24 chromosomes in chimps and you make that chimp a diploid, you go to 48. You take the 23 chromosomes you had in humans and make it diploid, and you've gone down to 46. So we do have remarkable similarities in our DNA sequences between chimps and humans. It's just that we combined 2 chimpanzee chromosomes into 1 somewhere along our human development. Pretty neat story, I think. So those are the chromosomal rearrangement mutations. They result in large-scale changes in chromosome morphology and chromosome structure. They are the 4 classes: duplications, deletions, inversions, and translocations. Now we're going to move on to the changes of chromosome number, and we're going to start with aneuploidy. Aneuploidy is simply defined as the loss or gain of a single chromosome or a small number of chromosomes. It can happen if a chromosome is lost during mitosis or meiosis because of the loss of a centromere, because of one of those strange translocation events we just went over, because of the lack of the ability of cohesin to degrade. Just said that. I just said that. All of these result in the loss or gain of a chromosome. When we have the failure of sister chromatids to separate, we call that non-disjunction, the ability of chromosomes to disjoin. Non-disjunction is specifically for a cohesin failure, a sister chromatid separation failure, but any of these kind of things, these incorrect occurrences, can result in aneuploidy. There are 4 types of aneuploidy. Their names help you remember what they are. Nullisomy. Null in science always means nothing or none. So nullisomy is when you have no more members of that chromosome in your genome. In other words, both members of the homologous pair are lost. And so if you've lost both members, you've had a reduction of your chromosome number by 2. And for a human being, this would be from 46 to 44. Monosomy, one chromosome is lost. Not both, just one. And so here we've gone from 46 to 45. We don't have disomy, because disomy would be 2 chromosomes, and that's the natural occurrence. That wouldn't be aneuploidy. Aneuploidy is when we've lost or gained some. Disomy is when we have 2 and we're supposed to have 2. So we go from monosomy to trisomy. Trisomy, or threesomy, is when we've gained a single chromosome. So we have one extra chromosome, we've gone from 46 to 47. And then there's tetrasomy, foursomy, where two chromosomes have been gained. And of course we've gone from 46 to 48. So we have nullisomy, a loss of 2, monosomy, a loss of 1, trisomy, a gain of 1, tetrasomy, a gain of 2. What are the effects of aneuploidy? Well, it's possible for an individual to have multiple instances of aneuploidy and still have 46 chromosomes. What? I just said that it was gain or but stop and think about it for a second. Imagine you have monosomy of one chromosome pair and trisomy of another. Well, the trisomy is going to give you one extra, the monosomy is going to give you one less. You balance out to 46 total. But are you healthy? No. No, because your gene dosage is completely screwed up. It's also possible to be double trisomic, where you have one extra chromosome for two different pairs. And so you have 48 total, but you're not tetrasomy. In any event, regardless of how you slice it, aneuploidy usually drastically affects the phenotype of the individual, and in almost all cases, aneuploidy is lethal, in almost all species. This is due to a complete imbalance of gene dosage, changes in the dosage of multiple genes across that entire chromosome that has either been lost or gained. There is an exception to this, however. The exception to this almost complete lethality is the X chromosome. In humans, especially in females, trisomy of the X chromosome is tolerated quite well. We said this in our sex determination lecture. In fact, most trisomic women for the X chromosome are living life completely undiagnosed. They don't even know they have trisomy X. Why do you think that is? I want you to think long and hard about that. I'm not going to define it for you. I want you to think about that as this lecture goes on. We also have Y aneuploids. Males who are aneuploid for Y, they have more than one Y, are usually viable because the Y chromosome is so useless. I mean, you have SRY, which initiates testis formation, and that's about it. So you have too many of nothing but SRY, it's not going to hurt you so much. But that's for the sex chromosomes. For the autosomal chromosomes, aneuploidy is a pretty big deal. Believe it or not, about 30% of all successful fertilizations or conceptions in humans spontaneously and naturally abort via miscarriage. Most so early that the mother rarely even misses a period. So most of the time the mother doesn't even realize she was pregnant. She might be a little bit late. She might have a heavier period than normal, but she doesn't realize that she actually had a fertilized cell in her body. 50% of these miscarried fetuses have aborted or miscarried because of chromosomal mutations, most of them being aneuploidy. In humans, only about 2% of fertilized conceived aneuploids survive until birth. And believe it or not, this rate of chromosomal defects is much, much higher than any other animal studied. Most animals have very little aneuploidy at all, and almost all of those aneuploids are not born. In mice, about 2% of all conceptions are aneuploid total. 2% of all conceptions are aneuploid, whereas for us, about 30% of conceptions are aneuploid. What this really means is, as a species, we suck at getting pregnant, and more importantly, we suck at meiosis. We're really, really too complex for our own good, and we've gotten so many genes and chromosomes doing so much that we're just not very good at efficiently separating them into gametes. Most human aneuploids are suffering from X or Y chromosome aneuploidy for obvious reasons, because these are the most tolerated, these have the least symptoms, and so obviously these are going to be the most viable. I already said it, but most autosomal aneuploids are spontaneously aborted. It makes sense, but since smaller chromosomes have fewer genes, and gene dosage is really our issue here with aneuploidy, the smaller chromosomes tend to tolerate aneuploidy better because you're doing without or having too many less genes than if you were dealing with chromosome 1, for instance. So what's the most famous trisomy? The trisomy you've probably already encountered. Trisomy 21. What does that really mean in the context of this lecture? Well, it means you're aneuploid, and the trisomy tells us you have 3 chromosomes instead of 2, so you have one too many chromosomes, and the one chromosome you have too many of is chromosome 21. Chromosome 21 in our karyotype is the second smallest chromosome pair. And so, stands to reason. Smaller chromosomes more tolerate trisomy or monosomy because you have fewer genes on them and the gene dosage balance is less skewed than it would be for a larger chromosome with more genes. We're all familiar with trisomy 21. I think we're all fairly familiar with Down syndrome. Many of us, myself included, might know someone close to us who has it, and so we won't go into too much detail there. However, there are other human trisomies that do result in viable children, in viable offspring that are more rare than Downs and probably trisomies that you haven't heard of. So we'll go through those in a little bit more detail because they should be unique and novel to you. Trisomy 18, so an extra chromosome 18, is also called Edwards syndrome. It's fairly rare. It occurs about once in every 8,000 live births. Babies are severely mentally handicapped. They tend to have low-set ears, a very short neck, and heart problems, so they're easily diagnosed upon visual or morphological changes. Few live for more than 1 year. This is an example of an individual with Edwards syndrome or trisomy 18. Trisomy 13, or Patau syndrome, even more rare, about twice as rare as Edwards syndrome, occurs about once in every 15,000 live births. Again, severe mental and cognitive defect, very small head, sloping forehead. Individuals tend to have a cleft lip and cleft palate. Sometimes we have polydactyly, extra fingers and extra toes. 50% of these individuals die in the first month of life. It's just so sad to talk about this stuff. 95% of those with Patau syndrome die by the 3rd year of age. And here is an individual with Patau syndrome. Trisomy 8, very rare trisomy on one of the larger chromosomes, exceedingly rare. It occurs about once in every 25 to 50,000 live births. Again, we have mental cognitive defects, although not as bad as Edward or Patau, and the characteristic physical alterations are present. It's easy to see trisomy 8 because those individuals are characteristically present themselves in their appearance. Trisomy 8 is so rare it doesn't have a separate name or syndrome name after it, and most individuals with trisomy 8 actually live a pretty normal life expectancy, so they make it to an average age. This is an example of an individual who's trisomy 8. So, what causes aneuploidy? I know in the mitosis and meiosis lecture way in the beginning of the semester, many of you said in your reflections, well, how is this possible? How can this even occur? What leads to problems in disjunction and in separation, in cystochromatid separation during meiosis? Well, most aneuploidies arise from non-disjunction, the failure of cystochromatids to separate during meiosis. Here's a little schematic of what we mean when we say that. We're supposed to line up in metaphase with homologous chromosomes side by side. This is after crossing over has occurred. And then during anaphase 1 in meiosis, we should be pulling homologous pairs apart from one another. What has happened here is that both light blue chromosomes of the same pair have been pulled to the same side of the cell. Does it happen often? No. Are there checks and checkpoints for measuring this, making sure it doesn't happen? Sure there are. Does it ever happen? Yes, it does. Mistakes occur during meiosis, and this is one of them. As these cells undergo meiosis 2 and cystochromatids separate, we can easily see that the two gametes arising from this cell are going to have 2 copies of the light blue chromosome each, and these two coming from this cell are going to have only 1 copy each of the dark blue chromosomes and none of the light blue. If these 2 eggs are fertilized, we are going to have a monosomy event, 1 chromosome too few. If these 2 eggs are fertilized, we're going to have a trisomy event, 1 chromosome too many. Since males are constantly producing new gametes and spermatogenesis is constantly occurring in males as long as testosterone levels can support it, but female gametes are kind of set early on in development. Yes, a female develops all of her eggs when she herself is still in utero, but actually those eggs are paused right at the end of prophase 1. So when a new egg is needed and when an egg needs to be matured, those female eggs are released from prophase 1 and allowed to continue on with meiosis. So females actually do develop their eggs as they develop into adulthood. But with all that being said, meiotic problems are observed more readily in females as their age increases because males are making so many sperm. Males are making sperm in such large numbers, and truth be told, male sperm are easy to make. I mean, you're talking about a DNA delivery capsule with a tail. That's male sperm. That's the male gamete. It's DNA in a hard shell with a tail on it. Can't really screw that up so easily. The female egg has to be so carefully prepared because of these asymmetrical cell divisions, creating polar bodies. You might remember that when we talked about oogenesis, polar body formation. And we have these, obviously, these two separation events in meiosis, so making a female egg is much trickier. And the female egg is made, the ovum, the fertilizable egg, is made in the developmental period that egg is needed. In other words, if a 35-year-old woman is making an egg, she prepared that egg while she was 35, not when she was in utero. That cell was established and prepared in utero, but it was paused at prophase 1. So what do we see? Well, we see with later age, especially in the mothers, we have a much, much higher prevalence of these non-disjunctive aneuploidy events occurring in conceived offspring. So 20-year-old mothers have about 1 in 2,000 chance of having a child with Down syndrome. Once you hit 30, it's about 1 in 900, so the risk has almost doubled. But from 30 to 40, it's gone up tenfold, from 1 in 100 to about 1 in 900 to 1 in 100. So once you hit 40, you've got about a 1% chance of having a child with Down syndrome. And then at 50 years old, 1 in 12. So almost another tenfold increase. Almost a 10% chance. All other factors taken off the table. About a 10% chance of having a child with Down syndrome if a mother is getting pregnant at age 50. Father's age, almost insignificant in its contribution to this. There's something called uniparental disomy. Well, what the hell are we talking about? Uniparental disomy translates into one parent giving 2 chromosomes. Right? Getting 2 chromosomes from one parent. Uniparental, one parent, disomy, giving 2 chromosomes. This is almost like a compensation effect. Sometimes a cell is conceived trisomatic, but it loses a chromosome early on in development, and so it does develop normally. If we have the loss of let's say the trisomy was coming from the mother. In other words, the mother had the non-disjunction event. If the chromosome that was lost was the paternal chromosome, we have a child born with no parental, no paternal chromosomes for that number. In other words, both of those chromosome 21s come from the mother. And the father contributed no chromosome 21 to that child because the father's chromosome 21 was lost. This is uniparental disomy. The child is disomatic, only has 2 chromosomes like it should, but both of those chromosomes came from one parent, which is strange. This child, though, will develop normally. There'll be no issues with this child's development at all because the child has the right number of chromosomes, and those chromosomes are healthy, coming from a healthy mother. So all of those are examples of aneuploidy. Now let's wrap the lecture up with polyploidy. What is polyploidy? Well, most eukaryotes are diploid, 2 of each chromosome, diploid, 2 chromosome sets, and they remain diploid for most of their lives. They get 1 chromosome set from one parent and 1 chromosome set from the other. If an entire set of chromosomes fails to separate during mitosis or meiosis, we can have a loss or gain of an entire chromosome set, depending on which cell we look at. That's polyploidy. Polyploidy is having more than 2 sets of chromosomes. We can have triploids, which is we have 3 sets, tetraploids, 4 sets, pentaploids, 5 sets, even higher. Polyploidy is quite common in plants. It's actually the major driving force behind plant speciation. However, not so common in mammals. Mammals are very intolerant of polyploidy because it screws up the gene dosage so much. About 40% of all flowering plants and between 70 and 80% of all grasses and most crop plants, wheat, oats, cotton, tobacco, etc., are polyploids. You can see that here. Potatoes are tetraploid. polyploidy 4N. They have 48 chromosomes. Bananas, triploid. Peanuts, tetraploid. Sweet potatoes are sixploid, whatever the prefix is for that. Tobacco is tetraploid. Cotton's tetraploid. Wheat and oats, sixploid. Sugarcane and strawberries, eightploid. Viable polyploids have also been found in one mammal. There's a rat in Argentina. This cute little guy right here is thought to be triploid, but there's some lack of consensus on that, some controversy on that. Some scientists actually think that that's not the case. It's still up in the air. We've never found a viable polyploid in birds, so it tends to be more of a plant effect in general. There are two types of polyploidy, autopolyploidy and allopolyploidy. When all the sets of the chromosomes are from a single species, we have autoploidy. These types arise from problems with mitotic divisions, problems with meiotic divisions. These are naturally occurring polyploids within a single species. When polyploidy represents a hybridization between different species, and this is where the plants get involved, with chromosome sets coming from different cells of different species, we have alloploidy. This can happen naturally, but we make this happen much more prevalently with agriculture. So most alloploids are sterile in the sense that they can't give rise to a new alloploid species, but they can self-fertilize often and keep their own propagation going with new seeds, and plants often self-fertilize. So these polyploids can perpetuate themselves by self-pollination and giving rise to their own seeds. But again, we do this with our breeding that we do with plants. We've constantly been trying to get plants to grow bigger and grow sweeter and develop more rapidly, and we do this by mixing the DNA of many plants together, creating artificial alloploids. Artificial in the sense that they wouldn't happen naturally, but still natural in the sense that we're not using genetic technology to do this. In the 1920s, I love this story just because I'm cynical. In the 1920s, there was a Russian biologist, and he realized that only the leaves of cabbage were edible, and the roots weren't, and only the roots of radishes were edible, and the leaves weren't. So he set out to do the obvious and create the Frankenstein plant that would be entirely edible. The Frankenstein plant that would have the leaves of the cabbage and the roots of the radishes, and the whole thing would be edible, and he did that by creating an alloploid. He mixed entire chromosome sets, and it worked. Believe it or not, it worked. He made this thing. This is what it looked like at the time. This is the paper he published providing this. The reason why we aren't all eating cabradishes or rabbages is because it worked the wrong way. He made the plant that had the leaves of the radishes and the roots of the cabbage, and so it was completely inedible. So why polyploidy, though? Why would this happen? How would this happen? Why would there be any natural mechanism for this at all? Well, typically polyploids are larger than normal diploids. See, the polyploidy, the excess DNA that the extra sets of chromosomes causes the nuclei of the cells to be bigger, to accommodate that extra DNA. Cell size correlates to nucleus size, so as nuclei get bigger, the cells get bigger too. Tissue and organ size, and then as a result organism size, always correlates to cell size. Larger organisms tend to have larger cells, and smaller cells give rise to smaller organisms. For this reason, and this reason alone in many ways, farmers have selected for polyploids over the centuries of agriculture because polyploids give larger plants, and larger plants give larger fruit and better yields. These farmers knew nothing about polyploidy, knew nothing about chromosome sets, knew nothing about DNA. They only knew that by forcing plants to do this, they got larger fruit. And so farmers forced plants to do that. But what they were really forcing on the molecular level was the maintenance of additional chromosome sets. Polyploidy also appears to be a driving force in overall evolution. If you think about it, much like the gene duplication story, giving a cell an entire new set of chromosomes gives it an entire new genome to tinker with at very low risk to the organism. We're not talking about a single hemoglobin gene. We're talking about a whole new freaking genome that you can play with and mutate and see what comes out of it. Yeast is actually a tetraploid. Baker's yeast, brewer's yeast, the yeast that we make beer and bread with, yeast is a tetraploid. And it's believed that this gene duplication event where the diploid yeast became a tetraploid yeast, where 2N went to 4N, right? A whole new diploid genome was maintained in yeast cells, allowed these yeast cells to completely mutate and tinker with an entire genome, its entire extra set of chromosomes. And by doing that, yeast evolved a completely novel metabolic strategy. Yeast can respire the same exact way we do. Yeast can take oxygen out of the air, mix it with sugar, and create large amounts of ATP. And the byproducts of that process are water and carbon dioxide. The same exact thing we do, yeast can do. But yeast also ferment. Yeast can make or break sugars down by fermenting those sugars and create ATP completely independent of oxygen. This is a pretty good evolutionary advantage, right? Make tons and tons and tons of ATP with oxygen just like we do. But then when there's no oxygen around and you and I suffocate and die, yeast just flips a switch and goes on fermenting and can make energy in the absence of oxygen too. It can do so because of the gene duplication event. And you knew yeast could do this. You knew yeast could respire and ferment. You've encountered it in your everyday lives. Yeast makes bread dough rise as a result of its respiring. The yeast releases carbon dioxide. The carbon dioxide proofs or puffs up the dough, making the bread light and airy. Yeast causes bread dough to rise as a direct result of respiration. But we've also experienced the wonderful effects of yeast fermenting. Yeast fermentation breaks sugars down and causes those sugars to become ethanol. And that ethanol is what's responsible for the brewing aspects of yeast. That's yeast fermenting. But yeast would have never had the opportunity to evolve this entire new metabolic strategy if not for the ability to tinker with an entire genome. This takes more than 1 or 2 gene duplications. In order to have a new and novel way of harnessing energy, you need an entire genome, an entire set of genes to tinker with. And if we look at the Saccharomyces cerevisiae lineage, that's baker's yeast or brewer's yeast, we see that yeast started with one set of genes, one set of chromosomes, but then with 2 sets could tinker with or change some of these. You see, on one of its sets of chromosomes, this gene has been completely inactivated, but it's been mutated or changed in the other. Here, the first gene has been completely deactivated through mutation, but the second gene has been changed. But sometimes, both genes on both sets of chromosomes was maintained and tinkered and changed in different ways. Yeast's closest relative, called K. lactis, it's a different type of yeast, did not have the gene duplication event and still is only diploid, and this K. lactis cannot ferment and respire. It is restricted to only one metabolic strategy because it didn't have the opportunity to tinker and play with a genome. Even in our own evolution lineage, there have been events of polyploidy. Even in our own, way, way, way back when, prior to the primate line, there have been polyploidy events that have allowed us to evolve our own multiple strategies for survival. So what did we talk about in this lecture? Well, we began just talking about general chromosome morphology. What do chromosomes look like? How do we describe chromosomes? What terms do we use when describing chromosomes? And how do we recognize chromosomes? We use terms like metacentric, submetacentric, acrocentric, and telocentric to describe the position of the centromere. We talked about staining patterns as ways of visualizing chromosomes. And then we did all this. And I'm actually going to leave the summary with this because Table 9.3 from your textbook does a marvelous job of taking this entire longer than average lecture and condensing it down into a single table. We talked about all the different chromosomal rearrangements, duplications, deletions, inversions, translocations, and we defined what those different chromosomal rearrangements were. We talked about the different subtypes of aneuploidy, nullosomy, monosomy, trisomy, and tetrasomy, and defined what those were. And we even defined the different cases of polyploidy, autopolyploidy, and allopolyploidy, and let you know what those were from. So that's it for this lecture. The next lecture will be on the genetic workings of bacterial cells. That's the first half of Chapter 8 from your book. We'll change gears once again and start talking about how these model organisms function at the genetic level, but we'll save that for the next lecture. And until then, thanks for watching.